Assalamu alaikum warahmatullahi wabarakatuh. Another embryology lecture, and we will carry on with the development of the fetal membranes. I'm Dr. Dalia Saleh, professor and head of anatomy department at Mansoura University, Egypt. In today's lecture, we will discuss the following objectives. We will carry on with the fetal membranes. You have already studied the amnion and the umbilical cord, so we will study in this lecture the development of the yolk sac, the allantois, the chorion, and I will talk about the placenta, its formation and its function, and finally we will talk about the twins. First, let's see how the extra-embryonic cavities develop. If we look at this sample picture, this is the endometrium of the uterus. The outer trophoblastic layer is formed of two layers, cytotrophoblast and syncytial trophoblast, and the inner cell mass is formed of two layers, epiblast and hypoblast. Near the hypoblast there is a cavity that develops that is important for transmission of the nutrition to the inner cell mass. It is the blastocele. Then here implantation took place. We can see another cavity develops inside the epiblastic cells to provide it with its nutrition. This is the beginning of formation of the amniotic cavity. With completion of the implantation, we can see the beginning of the appearance of a membrane that lines the hypoplastic layer. This is the Husser's membrane, which transforms now the blastocele into what is called primary yolk sac. And if we look at the amniotic cavity, it is lined by amnioblasts. In this stage, a layer of mesoderm develops between the trophoblast and the Husser's membrane. It is called the extra-embryonic mesoderm. Since it lies outside the inner cell mass, it is called extra-embryonic mesoderm. Later on, cavities appear in the extra-embryonic mesoderm. These cavities will coalesce together and form a large cavity. This is the extra-embryonic coelom. And the extra-embryonic coelom will split the extra-embryonic mesoderm into two layers. A layer that lines the trophoblast and also covers the amniotic cavity. This is the somatic layer of extra-embryonic mesoderm. And a layer that covers the yolk sac. This is the splanchnic layer of extra-embryonic mesoderm. Now, the primary yolk sac will change its name into secondary yolk sac. So, you can see that we have three cavities that lie outside the embryo. The amniotic cavity lies dorsal to the embryonic disc. Secondary yolk sac lies ventral, and the extra-embryonic coelom is the outermost one. The yolk sac, and let's see how it develops. Let's revise this picture again. Here you can see two cavities. The amniotic cavity develops dorsal to the epiblast, and we have the blastocele lies ventral to the hypoplastic layer. But when the Husser's membrane starts to develop from the edges of the hypoblast, the blastocele will change its name into primary yolk sac. Then the extra-embryonic mesoderm is formed here, and cavities appear within this extra-embryonic mesoderm that will coalesce together and form the extra-embryonic coelom. The extra-embryonic coelom will split the extra-embryonic mesoderm into somatic layer and splanchnic layer. You can see that the splanchnic layer surrounds the primary yolk sac, and it will change its name now into secondary yolk sac. Then the yolk sac is also affected by folding of the embryo that takes place in the 4th week of development. Remember that the amnion grows and expand. In the same time, the yolk sac gets smaller. So part of the yolk sac will be trapped inside the body of the embryo and form the primitive gut. The rest of the yolk sac will lie outside the embryo. We call it the definitive yolk sac. And both the primitive gut and the definitive yolk sac will be connected by a duct. We call it the vitello-intestinal duct. With development, the vitello-intestinal duct elongates and the definitive yolk sac gets smaller and smaller. So you can see that the part of the yolk sac that already entered inside the embryo will form the gastrointestinal tract. The vitello-intestinal duct will pass inside the umbilical cord, and the definitive yolk sac will lie outside the embryo near the placenta. So what's the function of the yolk sac? It facilitates the transfer of nutrients from the developing trophoblast to the embryo. It is also the center of blood formation in early embryonic life. It forms part of the digestive tube, and it is the source of the primordial germ cells that will later on share in the formation of the gonads. I'll talk about the allantois. The allantois is a small diverticulum or small extension that projects from the caudal end of the yolk sac into the connecting stalk. After folding, the allantois and the connecting stalk move into the ventral aspect of the embryo, and in the same time a sac develops as a result of movement of the allantois ventrally. We call it the cloaca. And this cloaca or this sac ends at a membrane called the cloacal membrane. So in summary, the allantois is a small diverticulum that extends from the caudal end of the secondary yolk sac into the connecting stalk. It is the structural base for formation of the umbilical cord. It will form a part of the urinary bladder, and the blood vessels develop around the allantois will form later on the umbilical vessels. The second membrane we'll talk about is the chorion. The chorion or the chorionic plate is formed of the trophoblast and the somatic layer of the extra-embryonic mesoderm, and it is the outermost covering of the embryo. It encloses both the embryo and also the other membranes like the amnion and the yolk sac and the allantois. Later on, the amniotic cavity grows more and more as a result of folding on the expense of the chorionic cavity that lead to its obliteration later on. For the chorionic villi, we have three types of it, or three stages in its development. First, the primary stem villus. It's formed of cytotrophoblast and syncytial trophoblasts. So this is the cytotrophoblast form the inner core and surrounded by the syncytial trophoblast. The secondary stem villus develops when the extra-embryonic mesoderm appear. So it is formed of a core of extra-embryonic mesoderm surrounded by cytotrophoblasts, and we have the syncytial trophoblast at the outside. Then we have the tertiary stem villus. It develops when the extra-embryonic mesoderm differentiate into connective tissue and the blood vessels. So we can see the extra-embryonic mesoderm surrounded by the cytotrophoblast and syncytial trophoblast. And you can see that inside the extraembryonic mesoderm, the blood vessels develop. Let me remind you first by some events that took place early on during pregnancy. This is the blastocyst. You can see the zona pellucida. Inside it lies the blastula or blastocyst. It's formed of two layers, outer cell layer and inner cell mass, and you can see a cavity there. It is called plastocele. After hatching and degeneration of the zona pellucida, the blastocyst comes into contact with the endometrial lining of the uterus at the site of implantation, and the outer cell layer differentiates into two distinct layers: outer syncytial trophoblastic layer and the inner cytotrophoblastic layer. The syncytial trophoblastic layer proliferate and sends finger-like projections and erode the endometrium, and then the blastula will migrate inside the endometrial lining of the uterus for its implantation. In the same time, you can see that the inner cell mass now differentiates into bilaminar germ layer. Here you can also see the blood vessels of the endometrium and also the endometrial glands. Then the blastocyst migrates more and more deep into the endometrium, and the entry site is covered by what is called fibrin plug. Also, you can see that the syncytial trophoblast comes into contact with the endometrial blood vessels and also with the endometrial glands, and they will erode these structures. Then lacunae or cavities will appear between the cells of the syncytial trophoblast. These lacunae will be filled with maternal blood from the eroded endometrial capillaries and also from secretions from the endometrial glands. And this will provide nutrition for the developing embryo at this early stage. At the same time, you can see that the inner cell mass now differentiates into bilaminar germ layer, epiblast and hypoblast, and we also have two cavities here, amniotic cavity and yolk sac cavity, and another tissue appears outside the embryo between the amniotic sac and the yolk sac and the cytotrophoblast. It is called the extraembryonic mesoderm. Inside the extraembryonic mesoderm, a cavity develops. It is called the extraembryonic coelom or chorionic cavity. The extraembryonic cavity enlarges, and we can notice that the cytotrophoblastic layer begins to send finger-like projections into the syncytial trophoblast. These projections will form what is called chorionic villi. So if we take this area and enlarge it to see how the chorionic villi develop, let's see it in this picture. This is the syncytial trophoblast. This is the cytotrophoblastic layer, and this is the extraembryonic mesoderm. So when the cytotrophoblastic layer sends finger-like projections into the syncytial trophoblast, this is the appearance of what is called primary chorionic villi. Then, when the extraembryonic mesoderm invade these finger-like projections, now the secondary chorionic villi are formed. When blood vessels are formed inside this extraembryonic mesoderm and invade the core of the chorionic villi, now the tertiary chorionic villi are formed. So here is the tertiary chorionic villi. You can see it is formed of an outer covering of syncytial trophoblast. then a layer of cytotrophoblast, then a core of extra-embryonic mesoderm containing the blood vessels. You can also notice that the cytotrophoblastic layer sends column of cells that will make a shell. It's called cytotrophoblastic shell, which lies close to the endometrial lining of the uterus. Here we can see inside the endometrial lining of the uterus, and this area will be the decidua bizalis. We can see the endometrial capillaries or blood vessels that are eroded and fill the gaps between the chorionic villi. This is called the intervillous spaces. Let's go back to the chorionic villi. These chorionic villi at the embryonic pool will grow and form chorion frondosum that faces the uterine wall, while chorionic villi at the embryonic pool or away from the embryo will degenerate and form what's called chorion leaf, and this part of the chorion will bulge into the uterine cavity. Remember that the endometrial lining of the pregnant uterus is called the decidua. So the part of the decidua that comes into contact with the chorion frondosum, we call it the decidua bizalis, while the decidua that covers the embryo or the gestational sac, we call it the decidua capsularis, while the decidua that lies the rest of the uterine cavity, we call it the decidua parietalis. So, with further development and enlargement of the gestational sac, the decidua capsularis will overstretch and degenerate, and finally the chorion leaf will come into contact with the decidua parietalis, leading to obliteration of the uterine cavity. Placenta is made of the following: fetal portion made by the chorion frondosum, and a maternal portion is made by the decidua bizalis. The fetal side is limited by the chorionic plate. The maternal side is limited by a plate called the decidual plate, which forms septa. These septa will divide the placenta into pieces. We call it cotyledons. The spaces between the septa, we call it the intervillous spaces, and these intervillous spaces will communicate with each other below these septa. So what is meant by placental barrier? It is the membrane that lies between the fetal and maternal blood. Remember that there is no mix of blood between the mother and the fetus. So this membrane or barrier is formed of the following: the endothelial lining of the villus capillaris, the mesodermal core which lies inside the tertiary villi, the cytotrophoblastic layer, and the syncytiotrophoblastic layer. With further development and the maturation of the placenta, the cytotrophoblastic layer will disappear, and with further maturation, the mesodermal core of the villi will also disappear. So the placental barrier now at the full-term placenta is formed by the endothelial lining of the villus capillaris and the syncytiotrophoblastic. So what is the structure of the full-term placenta? It is rounded in shape and disc-like. That's why in Greek it is called plakos or flat cake. It's about 15 to 20 centimeter in diameter, 2 to 3 centimeter in thickness, nearly half kilogram in weight, and has two surfaces, a maternal surface and a fetal surface. This is the maternal surface. It has a cobblestone appearance because of the shape of the cotyledons that are separated from each other by grooves, while this is the fetal surface of the placenta. It is smooth and shiny, covered with amnion, and you can see the umbilical cord attached to its center. Also, you can see the umbilical vessels and the chorionic vessels clearly seen under this thin amniotic membrane. For the fetal circulation, we can see here the maternal side, or the decidua physalis, containing the maternal vessels, and we can see here the intervillous spaces containing the tertiary chorionic villi, and inside it lies the fetal blood vessels. So blood come in the maternal arteries and fill the intervillous spaces. If we enlarge it, oxygen will move or pass in this thin placental barrier from the area of high concentration to the area of low concentration, meaning that from the mother to the fetus. And the carbon dioxide will move back from the fetus to the mother. The non-oxygenated blood or less oxygenated blood will be picked up by the maternal vein back to the mother circulation. Remember that no mix of blood between the mother and the fetus. So what are the functions of the placenta? The placenta acts as a tool or a mean for exchange of material between the mother and the fetus. It acts like a defense barrier. It produces hormones like human chorionic gonadotropin, progesterone, and estrogen. Now let's move to the final topic, which is the twins. By definition, twins is the delivery of two babies. Its incidence is about 1 in each 85 pregnancies. The outcome will be a twin. Many factors may cause having twins, either hereditary, meaning that it runs in families, or it may be caused by stimulation of the ovary by ovulating drugs, or as a result of in vitro fertilization. There are two types of twins, either dizygotic or monozygotic. The dizygotic twins are also called biovular twins, or non-identical twins, or fraternal twins. It's about 70% of the twins are dizygotic, and it results from fertilization of two ova by two sperms. The twins are of the same or different sex. Genetically, they are non-identical, like brothers and sisters, and the twins have different fingerprints. So, if there are two ova that are ovulated or liberated from the ovary in the same time at the same month, and they are picked up by the uterine tube, and they get fertilized by two different sperms, each will develop into a zygote, and then cleavage will start by giving 2 cell stage, 4 cell stage, 8, 16, and 32 cell stages, or in this case we call it the morula. The two morulae will travel inside the uterus, and each will implant and develop into a separate embryos, as we can see here. So these dizygotic twins each has his own placenta, his own chorion, and his own amnion. While the monozygotic twins are called uniovular twins, or identical twins, or two twins. It's about 30% of the twins are identical, and it is caused by fertilization of one ova by one sperm. There are three types of the monozygotic twins. If the separation occurs at the two-cell stage, or if the separation occurs at the early blastocyst stage, meaning that at the inner cell mass, or if the separation occurs at the late blastocyst stage or in the bilaminar plate. So the criteria of these monozygotic twins, they are of the same sex, genetically are identical, they carry the same physical character, and they also carry the same fingerprints. So if the separation was at the two-cell stage, as in this case, so each of these two cells will develop into a separate embryo. So each embryo will have his own placenta, his own chorion, and his own amnion. If the separation occurs later on, so you can see here the morula that will develop into a blastocyst with inner cell mass and blastocyst cavity. But in this case, the inner cell mass separates. So each group of cells will develop into a different embryo. So the resulting twins will have one placenta. They share in the same chorion, but each will have his own amniotic cavity. If the separation occurs at the bilaminar disc stage, so again this is the morula that will develop into a blastocyst, which has here an inner cell mass that later on will differentiate into a bilaminar germ disc. So in this case, there is separation at the bilaminar germ disc. So the separation occurs later on. So the resulting twins will have one placenta, one chorion, and also one amniotic cavity. So in summary, the early is the separation, the more independent the embryos are. So if there is separation at the two-cell stage, so each embryo will have his own amnion, his own chorion, his own placenta. They do not share in anything. But the late separation, as in the bilaminar germ layer, they share in everything. They share the same placenta, the same chorion, and the same amnion. Another type of twins called conjoined twins. This occurs when there is a splitting of the zygote at late stages, and this separation may be incomplete. So the resulting embryos are united and they share the same organs. So if the fusion at the thorax, we call it the thoracopagus twins. If the fusion was at the head region, we call it craniopagus. If the fusion was at the sacral region, we call it pyopagus. The twins may be united to each other by skin bridge or even share the same organs like the liver, the brain, or the heart. What's the complication of having a twins? Twin pregnancies have high incidence of prenatal death, preterm delivery. They tend to be smaller in weight at birth, and sometimes there is a syndrome called twin transfusion syndrome, in which one of the two embryos will get more blood supply from his brother or sister. So one of them will be small in size and the other one will be large in size. As we can see here, this baby is pale and smaller in size in comparison to his twin brother. This will be the end of my lecture. Thanks for listening and see you in the tutor availability. Thank you.